Hi everyone. So this video, I'm not going to lie, it is an amendment to the other videos in this unit. What I mean by that is this information was added after I recorded all of the videos. However, I think it is a good idea to keep this as a separate video, as a separate topic, because this topic will be reoccurring throughout this year in biology. So what we are going to be talking about is something called an enzyme. Now, what we've been talking about are different macromolecules. So very large molecules. Specifically, we've been talking about carbohydrates, lipids, proteins, and nucleic acids. This is not like an extra macromolecule. However, it is part of the class of proteins for macromolecules. So just like how proteins have a very specific job inside of your body, so each specific protein has its own very specific job. Kind of think about it like a lock and key model. There's only one key that opens one lock, they fit together. Very similar in this scenario are enzymes. So enzymes will have very specific jobs in the body, and each enzyme does a different job. So their actual job in the body is to put molecules together or to take molecules apart. And they also will speed up a chemical reaction by lowering the activation energy. I'm going to come back to this third point here on another slide. And for now, I'm just going to focus on these two functions of an enzyme. So like I said, the enzyme's job is to either take a molecule and break it apart into two different molecules, or it's taking two molecules and trying to put it together into one molecule. And there's many different enzymes in your body. Each specific enzyme will work on a very specific molecule. So that's what I mean by a lock and key model. If you imagine the enzyme as the lock and the molecule that it's working on as the key, it only one of them fits together with the other one. So that would be the basic job of an enzyme. Now, I want to come down to this diagram right here because this will show us how an enzyme works. I do have a video coming up as well that's animated so we can see what this looks like, but just go with me for a second. So this gray blob looking thing is going to be your enzyme molecule. This green thing up here is the molecule that the enzyme is doing its job on. So this molecule right here is called a substrate. Now, like I said, the substrate is the molecule or molecules that the enzyme is working on. So what's going to happen is that the substrate up here is going to bind to the enzyme's active site. That's what you're seeing right here in this part of the diagram. So where the substrate binds to the enzyme is what we call the active site. That's this definition right here. So the molecule or the substrate fits directly into the enzyme and they fit together at the active site. Now, what the enzyme does is it will lower the activation energy and it will cause this molecule to break apart into two different pieces, which is what you're seeing right here. So we went from this green molecule as one substrate, and we broke it into two separate molecules, which creates two separate products. So a product is the molecule or molecules that the enzyme makes. I do want to note that in this process, in order to break the molecule apart, the enzyme does have to change shape. However, at the end of this process, the enzyme does revert back to its original shape, and so it can be recycled and reused multiple times. Now, like I said, an enzyme can break a molecule apart, and that's what we just looked at, but this can also go in the opposite direction as well. So it could take these two molecules and put them together into one molecule. So we could just flip these arrows around in the other direction, and an enzyme could do that job too, and then we would call these the substrates and this the product if that's what was happening. So let's look at this activation energy I've been referring to. And basically, like I said, in order for a chemical reaction to happen, it needs a certain amount of energy put into it. So without an enzyme, like I said, the enzyme lowers the activation energy. Without the enzyme, this chemical reaction could take years, like longer than your lifespan in years for this reaction to happen. However, the enzyme makes it work like that. So when we look at this graph right here, if these are the reactants, so that's the molecules that we start with, and these are the products, this is the molecules that we're making or ending with, we, as the reactants for this chemical reaction, we have to build some energy, build. And right here is the activation energy. Okay, so once we reach that energy, that's when the chemical reaction can happen and we can drop some energy. It's usually released as heat, and we are left with our products. Now, with an enzyme, what the enzyme does is it lowers the energy needed for this chemical reaction to happen. So we still have to build some energy, but we don't have to build as much energy. And so this makes it a lot easier for the chemical reaction to take place. And keep in mind, a catalyst is just a chemistry word that kind of is talking about an enzyme. An enzyme is a biological catalyst. So they're both lowering the activation energy. The enzyme will do it inside of our bodies because we are living things. So that's what I mean by lowering the rate of activation energy. But because we don't need as much energy, the reaction happens a lot quicker. So this is why we're able to, I guess, like live and react in the speed that we do. There are a few factors that can affect how well your enzymes are working inside your body. So keep in mind, if your enzymes are not working properly, you die. You need your enzymes to be doing their specific jobs inside of your body in order to stay alive. So there are three things, like I said, that will affect the enzyme activity, how well the enzyme can do its job. And we're going to look at it in these three graphs over here. So the first thing that can affect how well an enzyme can do its job is temperature. This is in degrees Celsius. I know that we're used to using Fahrenheit, but it works very similarly. Keep in mind that the body's temperature in Celsius is 37 degrees Celsius. We know it as 98.6 degrees Fahrenheit. So 37 degrees Celsius is equal to 96.8 degrees, I'm sorry, 98.6 degrees Fahrenheit. They're equal. So what we're seeing here is for this graph, we're looking at temperature on the x-axis down here, and we're looking at enzyme activity on the y-axis up here. So that means the higher that the red line is, the more enzyme activity there is, the better able the enzyme is to do its job. So we do want to see this line going as high as possible. So looking at this graph right here, what this shows us is that at very cold temperatures and at very hot temperatures, the enzyme cannot do its job. The enzyme would denature. So remember that an enzyme is a protein, and when a protein gets heated up or cooled down, it can fall apart. It no longer works, and it's called denaturing. So as we go up in temperature, we can see that the enzyme activity increases as well. So this enzyme likes to work at a slightly higher temperature. It peaks around this point right here, which actually is right around 37 degrees Celsius. So this would be an enzyme that we find inside of our bodies and works best inside of that environment. After we reach that temperature where the enzyme works best at, then we see a very steep decline in enzyme activity. For this graph over here, we're looking at pH. So remember, pH is a way that we can measure how acidic an environment is or how basic an environment is. We like to keep the body at a pH of 7, a neutral pH. And again, that's kind of what we're seeing here in this graph. So again, we have pH across the x-axis, across the bottom. And then we have enzyme activity going again on the y-axis. And so as the red line goes higher, that's better. We're seeing more enzyme activity. So same idea. We start off with a really low pH, a really acidic, I clicked something, a really acidic environment. And as we slowly climb up the pH scale, as we slowly make it more towards neutral, keep in mind we're not at neutral right now. This is when enzyme activity peaks. So that's right around a 5. So it's still slightly acidic, but it is closer to neutral. So this specific enzyme probably is found like in our gut. right after stuff leaves the stomach because our stomach is very acidic. It could also be found maybe in the blood. Our blood tends to be slightly acidic as well. But the idea here is that enzyme activity peaks around a pH of 5, and then after that, again, you see a very steep decline in enzyme activity. So both of these graphs, I call them a Goldilocks graph because you want it not too hot, not too cold, just right. So again, not too acidic, not too basic, just right. And so that's what you see in both of these graphs. Now, this graph is slightly different than the other graphs. We're going to be talking about substrate concentration. So remember, a substrate is the molecule that the enzyme does its job on. It's the molecule that the enzyme works with. So the idea here is that as we go along the x-axis, we are adding more substrate to the environment. So we're adding more molecules for the enzyme to do its job on. And again, along the y-axis, we're looking at enzyme activity, so how well the enzyme can do its job. So what we're seeing here is a different looking graph. As we add more substrate to the environment, we see the enzyme activity going up, 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 and then it sort of peaks, and we get a nice plateau on this graph right here. So let's think about why we're seeing this, right? So you'd think that the more substrate we add, the more stuff an enzyme has to do, the faster it's going to work, the better it's going to work. Let's compare this to ourselves, okay? Or you can compare it to a car too. I'll do ourselves. So let's say that I give you a job of folding paper airplanes, and I give you a new piece of paper every 15 minutes. Generally speaking, folding a paper airplane takes maybe like 30 seconds. I'll give it 30 seconds for this example. So if you're given 15 minutes to do a job you can do in 30 seconds, you're going to work at a pretty slow pace if you're only getting a new paper every 15 minutes, okay? So when in this scenario, I now give you 2 pieces of paper to fold in 15 minutes, you've now doubled your productivity because, again, the paper airplane takes 30 seconds, so you're working for a minute, and then you have 15 minutes to wait before your next piece of paper comes. So you are able to do your job better, but still not great. Now, as I add more and more paper to those 15-minute intervals, you can start folding more and more paper airplanes. However, once you are given 30 pieces of paper, because remember, I'm saying you're folding your paper airplane in 30-second intervals, so if you divide that by 15 minutes, it would be 30 paper airplanes. Once I give you 30 pieces of paper to fold in 15 minutes, that's your max working capacity. You can't work any faster. So that's what we're seeing up here with the plateau. It's, yes, we keep adding more and more substrate so the enzyme can do its job, but at some point, the enzyme can only work so fast. It's the same idea as the gas pedal in your car. So I know some of you can't drive, which is okay. Hopefully you still understand the concept of a gas pedal. But it's sort of like the harder you push down on the gas pedal, the faster your car is going to go. However, at some point, you've pushed the gas pedal down as far as you possibly can go. The car has sped up as fast as possible as the car can go, and it won't get any faster. And so again, you'd see this plateau right here. So it's the same idea working with an enzyme. Before we go into the video, I did forget to mention one thing, just as a hint. So for MCAS or for anything we're doing in biology, if you see this suffix ACE, A-S-E, at the end of a word, it is referring to an enzyme. So in your head, you should be thinking, okay, whatever name it is, like helicase, ligase, amylase, DNA polymerase, RNA synthase, or ATP synthase. I obviously just listed a bunch of enzymes. You don't know what they do, but because you know it's an enzyme from the ASE, you know that these molecules are an enzyme, which means they are putting molecules together, taking them apart, and speeding up the chemical reaction by lowering the activation energy. So that's just a little helpful hint as you're reading through stuff. If you see that ACE, you know it's an enzyme, and then you should know what an enzyme does. So I do have a video I would like to play for you so that you can see what an enzyme looks like while it's doing its job.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
All right, so I know that was like a quick little video, but hopefully it gave you a good visual reference of what this might look like inside your body. We will get some more practice with this, but like I said, this is an important type of molecule that will keep coming up over the course of this year. So make sure you check in with your vocab notebook and you are filling out your slide for enzymes.